thank the organizers and Dr. Waxman and the other organizers for this great invitation to come here to this conference, which I love, and to come back to China, which I also love to do with my wife. So thank you very much. I will talk today about therapy. I will allude to some things that I'm very excited about in therapy, but I wanted to give some basic background that might do well for this first session, tell you some about our lab's work, but others as well in this arena, and then go into the translational implications at the end. So, this is a very nice review written recently by my colleague Peter Jones, and it emphasizes one thing in what we beginning to understand, I think, the biology of the epigenome, which is so important to any therapeutic implications. So we've all in this field, when I started 25 years ago in epigenetics, we've all been very promoter-centric, if you will. We've all worked on DNA methylation and chromatin changes around the promoter. But I know many of you in the audience have been reading the Nature papers out of ENCODE recently and understanding how we're evolving a much broader look at the epigenome. We know we have to understand chromatin and DNA methylation now in enhancers that are distal from the promoter. We need to understand the proteins that set up boundaries between translationally repressive chromatin and translationally active chromatin. We know, for example, that we not only need to understand canonical gene transcription, but non-coding RNAs that come in antisense directions or from non-genic regions of the genome. We're going to hear about those at this meeting. They're all of this is extremely important to coming to understand how we're going to think about epigenetic therapy. And if we look at a cartoon of some of the changes that take place in cancer, we know that, for example, DNA methylation at many sites where it ought to be present is lost in cancers, white lollipops. Many promoters that should not be methylated in their CpG island-rich regions at the promoters simultaneously in the same cancer cells gain DNA methylation. And we understand now from Peter Laird's work, from Andy Feinberg's work, and others with the deep sequencing of methylation, that these regions of losses and gains can be in the same megabase regions. That's very important because it says what's breaking down in cancer at many regions are boundaries between where methylation ought to be or ought not to be and chromatin changes. This becomes very important. The other exciting thing that's happened over the past several years is the recognition that many genes in cancer which encode for proteins that control the epigenome are mutated. And we're going to probably hear some about this. So enzymes can be mutated that put on the histone modifying marks. DNA methyltransferases can be inhibited, and enzymes that remove DNA methylation, we're going to hear, can be mutated. And the genes that move the nucleosomes around can be mutated. And we need to understand, so far we understand very little about what those mutations mean to the epigenome, but they're frequent enough they know that they must be important, and we need to consider all of this. Now, our lab was one of the labs that started with these gains of DNA methylation, which can be transcriptionally repressive in promoter regions. And in thinking about this in the whole context of everything, I will postulate to you that this is still a very important change because when this region is DNA methylated, that's a very dominant change for repressing gene transcription. So even if you were to activate enhancers or signal transduction that would try to drive this promoter, if it's in this configuration, that's still a very dominant change. So let me show you that selection for a DNA methylated gene. This is the recent paper on squamous cell, non-small cell from lung cancer from the TCGA consortium that was published, and it's on the P16 locus here where a deep study was done. So this is a locus, the RBP16 pathway and chromosome 9 for P16 that gets altered in about 72%, probably more, of squamous cell lung cancers. And most of the time it's due to something happening to P16. And this shows you a factor that for some driver genes about the epigenetic change. So P16 can be homozygously deleted, that's in the blue. It can be mutated in these tumors here, or, and the point I want to make, it's mutually exclusive in this case, the blue down triangles, it's DNA methylated. So the mutations are exclusive from DNA methylation. Furthermore, if RB is mutated, then P16 is overexpressed and not methylated. And here's the RNA-seq that goes with these data here. You can see homozygous deletions, obviously very low expression of P16, but if you've got one copy of P16 gone and one methylated, or both present but both methylated, the red circles show that the expression is almost low as completely losing the gene. If P16 is mutated, or RB is mutated, then the P16 expression is high. So this shows you that some genes can be selected for. But this Infinium plot from Illumina shows you the real world for the hundreds of genes that take on this change. And we understand far less about the nature of all those genes taking on this change and what they mean for the cancer. So on this Illumina plot, red is methylated, green is unmethylated, and these are all CpG island probes on this plot that should not be methylated in normal tissues. And that's shown right here because here's embryonic stem cells, adult stem cells, adult epithelial cells, different kinds of cells where the probes for these several hundred genes are all green. And you can see these cultured cancers all have hundreds of genes that are hypermethylated for DNA at their promoters. There's colon cancers, breasts on here. And if you take these same probes and now look at primary human tumors in the Cancer Genome Atlas Project, so you take the colon cancer probes, and you look at these hundreds of primary colon cancers here using the same platform. Here's the normal again, some noise in it, but you can easily recognize the normal, and you can see these genes methylated for hundreds of genes in the primary tumors as well. So I'm going to ask the question with you a little bit, is this a therapeutic target? But the basic biology of this is how does this happen, about which we're beginning to learn, and what does it mean for tumorigenesis, which is extremely important. So I'm going to show you briefly an approach we've used in the last couple of years to look at something about how these changes come about. It's work by Harry Esrawan in our lab, an informaticist, Leander von Nest, and Sarah Johnstone when she was in our laboratory, and others collaborating. But what we did was to take an adult stem cell system, mesenchymal stem cells, which by changing the calcium you can differentiate into bone progenitors, osteoblasts, and compare them to bone tumors. And then we did a profile using ChIP-seq to look at an active mark, H3K4 trimethyl, an inactive mark set up by Polycomb called H3K27 methylation. We'll come back to those. Matched these on Illumina to DNA methylation and looked on an array for gene expression. And what you see here is from this study, you can get out 380 genes that are all methylated in the bone tumors here, which are not methylated in the mesenchymal stem cells or the osteoblasts. And then you can look what kinds of chromatin marks these genes have when you compare them back to stem cells. And what we find is of those 380 genes, most of them in embryonic stem cells have both the activating mark of H3K4 methylation and the silencing mark at the promoter H3K27. That's what Brad Bernstein called bivalent chromatin, and we'll discuss that in a second. So when you summarize all of this, and you look back at these hundreds of genes, and then we also look from these different tumors, those genes, the majority of them, map back best to a state that they harbor in embryonic stem cells in the orange of bivalent chromatin. They also do that in adult stem cells, like hematopoietic stem cells. It's best back to bivalent chromatin. Now, what's important about bivalent chromatin, that's a state that has a poised low transcription associated with it. And it's thought that that helps keep the stem cells either as stem cells renewing, or it helps them not prematurely differentiate or commit to lineage. And you can see when these cells do, mesenchymal stem cells have already changed this to have mostly polycomb mark in the red, or the genes go to active genes as marked by the H3K4. The osteoblasts further change most of the genes to polycomb, and most of the methylated genes normally would just, some of them would have an active mark. Same thing, hematopoietic stem cells, normally these genes are not DNA methylated, remember that in the normal cells, convert many of these genes to polycomb alone and retain some as active with the green mark here. And most of the frequently DNA hypermethylated genes, again, map back to the highest 50 of all these tumor types, back to bivalent chromatin in embryonic and adult stem cells, and back to polycomb mark alone in progenitor cells. And this is very important because since they're not normally DNA methylated, somewhere in this transition during tumorigenesis, and we need to understand that transition between bivalency and polycomb and the genes that go from an inactive state to an active state, this probably is the key during tumorigenesis to at least understand where DNA methylation comes in and replaces those normal conversions between bivalency, active mark, and inactive mark. So I think the model is there. Is this important in tumors? I think it's extremely important. This is an Illumina for the TCH of about 250 primary breast cancers. And you can see the DNA methylation patterns here. This is the red, this is Chuck Bruce. These are triple negative tumors. And here are a bunch of genes that are hypermethylated in luminal, the more differentiated B in the black, or luminal A, and another group more intermediate here for luminal. And you can see that all of these genes that are frequently methylated here are not DNA methylated at the promoter and triple negative. And a number of labs have found that what's overdrive in these triple negatives seems to be the polycomb system. The enzymes for polycomb and their cooperative proteins are high. And so these patterns are very different for whether they have a lot of DNA methylation or not. And I would postulate to you that any epigenetic therapies that we apply in these settings, we have to understand these differences even in subgroups of tumors for these kinds of marks if we're going to wisely come in and learn how to treat them. What would you block here? What would you block here? May be very different. Or combinations might have to take these different kinds of biology into account. Most of the bivalent genes that abnormally get DNA hypermethylated in cancer, the ones that stay silent with polycomb normally but without DNA methylation, are mostly developmental genes, and this may be very important. And of those, they're mostly transcription factors and DNA binding genes. The bivalent genes that convert to active genes, for instance, in osteoblasts, for example, progenitor cells, are mostly in signal transduction pathways. And so we need to ask, what is it about these genes, or I think more groups of genes, which ones of these are important? If you can't re-express them because they're DNA methylated, which ones as groups may be important in driving the tumor? And we can look at this way. As I've told you, something seems to happen in tumor progression where genes that normally would either be bivalent or polycomb marked or have an active mark alone but not DNA methylated seem to, subgroups of them, take on DNA methylation, a tighter silencing. And the question we can ask, but we need to answer, is, does this help lock in, for example, the survival of primitive cell populations in cancer which continue to self-renew and or can't differentiate normally? I frame it as a question, and I think one in the field that we need to think about. I'll put it in another context, and that is, how do the epigenetic alterations in a more primitive type epigenome possibly interact with tumor suppressor genes and oncogenes? And this is Scott Lowe's hypothesis, or not hypothesis, is sort of proven. He calls the dual face of oncogenes. If you take the MYC gene and overexpress it in a normal, particularly differentiated cell, that cell sees it as almost like a DNA damage response. It's going to senesce or apoptose. But in the right background, MYC is obviously an oncogene and can addict the cell to oncogenesis. And one possibility is that these primitive epigenetic landscapes, if they're inherited in a cell population and see a MYC mutation or MYC overexpression or a RAS mutation, or increasing evidence that MYC can actually set up primitive epigenetic abnormal type settings, that you've got an epigenetic terrain underlying and helping these cells be addicted. And if you were to come in early with a prevention strategy or with a therapy strategy, the question is, could you take that epigenetic terrain away from those oncogenes or tumor suppressors and break the cell addiction? And I think this is a fundamental principle that possibly could underlie many of the therapeutic strategies that we might think about in present or the future. When do these changes arise? That's very important. And they can arise anywhere in tumor progression. But for many of the genes we're talking about, our lab, Jean-Pierre Issa's lab, a number of you probably in the audience know that you've worked on, that abnormal DNA methylation and chromatin changes often take place reasonably early in this Vogelstein-Kinsler furon model here. We know that polyps, for example, or even pre-polyp abnormal areas of the colon epithelium can harbor genes that are already DNA hypermethylated or have other changes, epigenetic changes that we're talking about. And so that, again, stresses this maybe understanding of stem progenitor-like cells in these early changes in cancer for these changes. How do they come about? Well, we're only learning how they come about. One thing I've told you is that the switch between polycomb and bivalency and these others can be altered such that DNA methylation comes in. But another thing is we're beginning to understand that risk factors for cancers, things that stress pre-tumor cells early in the progression of cancer, could be very important for setting up these chromatin changes and or DNA methylation changes. And I'm going to show you just a very quick snippet of work that we published about a little less than a year ago. Heather O'Hagan led this work in our lab, along with leadership from Wei Wang from China, who was a graduate student during these studies, and Subiji Sen and others in the lab. And what they looked at was a somewhat artificial, but I think important system. One of the things that happens, the leading risk state for cancer, perhaps, is chronic inflammation for many tumor types, including colon and others. And when you have chronic inflammation because of the cells that you bring in and the tumor cells themselves, they have to adjust to, and they are faced with increased reactive oxygen species levels. And so what they artificially did in this paper is to expose cancer cells to increased levels of what they used hydrogen peroxide and increased the levels of reactive oxygen species in these cells. And within 30 minutes, they saw the following changes that are really very important. So they did tiling studies. They marked the area of 8-oxoguanosine or other damage by phosphorylated H2X, mapped it along. This is chromosome 21 here. So a downward tick in the ratio means that H286 was depleted from this more pericentromeric or region of chromosome 21 and moved to the end of the chromosome, the more telomeric regions of the chromosome. And along with that, they saw moving and tightening to the chromatin and moving to this region, the DNA methyltransferase that does most of the maintenance methylation, DNA-MT1, along with SIRT1, a key histone deacetylase, along with another DNA methyltransferase, 3B. And what is this region of the telomere? And it looked like this on every chromosome. They went to the regions where were most GC rich and which harbored the genes in the more richly with the CPG islands we're just talking about. So that what I'm telling you is within a rapid time after exposure to this inflammatory-like buildup of an agent, complexes move because you can immunoprecipitate DNA-MT1, SIRT1, DNA-MT3B, and polycomb elements simultaneously from these regions. And you can see here's 8-oxoguanosine at the MYC promoter after damage, so the damage is here. That's a CPG island, the actin promoter, but a CPG-4 promoter gene doesn't get that kind of damage, at least measured by the 8-oxoguanosine. And you can see the changes in the individual genes. This is MYC. You see buildup of polycomb transiently within that 30 minutes in the red, a loss of the active H3K4 mark in the green, and decrease in acetylation of H4K16, and that would be SIRT1 coming into the picture. And if you take genes that have a low transcription, that aren't DNA methylated in these particular cancer cells, and you sequence, you do bisulfite sequencing, within that 30 minutes, seemingly almost the lower the basal transcription, you can see that individual alleles that are sequenced build up in the hydrogen peroxide treated cells over that spectrum in the untreated cells. And so if we had to look at 2012 and think about some things that affect the epigenome, I would say that we have Peter Laird's work showing that these vulnerable regions that both lose and gain the DNA methylation at the promoters seem to be vulnerable to this change. They harbor most of the polycomb bivalent marked genes in embryonic stem cells. And there have mutations now the best understood, perhaps, Ari Melnick's here has done work in this, is the TET mutations that allow DNA methylation to build up, and one of the regions it builds up in cells is in these genes. And so mutations and the changes in chromatin that accompany them are going to have to be considered in thinking about how these things come about. And then I've told you that risk states, assaulting these regions with DNA-damaging hydrogen peroxide-like reactive oxygen species may damage the chromatin in these regions, and if it lasts, if it doesn't snap back, all of this could be considered fundamental and just some of the things we need to think about in the cancer epigenome. So now let's talk for the remainder of the talk about therapy. So I was asked to give two talks. I was going to talk about lung cancer in this session, but I'm going to wait until Tuesday at 2 o'clock to tell you how excited I am about some approaches that our group in the Stand Up to Cancer in America is taking against non-small cell lung cancer that, if they hold up, will make a major change through, quote, epigenetic therapy and priming of subsequent therapies in this most deadly of cancers. And I'm going to talk about that in specific on Tuesday, but I'll allude to it here. So I keep showing this slide over and over again. I'll probably quit showing it because I'm looking for one thing. Here's the epigenetic therapist. This was in the economy. And here's this, I think, angry nucleosome. And every time I show it, I'm hoping for a switch in posture here, that these guys get on top and this guy drops. It doesn't look too good right now. But I think this is what we're hoping to be able to do in epigenetic therapy, is really stop the cancer process through mediating this. How are people doing it? You're going to hear about some of this. You heard a bit last night. So, for example, the repressive marks. DNA methylation put into the genome can't repress the transcription of anything until repressive chromatin forms around it. And where you put DNA methylation normally or abnormally in the genome is also influenced by the chromatin modifications. We heard a little last slide, if you reduce the demethylase for H3K4 methylation and build up this active mark H3K4, you actually can block, that would not be friendly to putting in DNA methylation. And so what pharmaceutical companies are doing, almost all the large ones have programs in this, they're doing excellent chemistry to block enzymes that write the histone modifications, that erase them, the demethylases we hear so much about, and the proteins that read these marks, the chromo and bromo domains, to do this. I'm going to talk about, this is a target, blocking the DNA methyltransferases to block methylation, perhaps, where it shouldn't be, or other facets of how these proteins function. You're going to see targeting in the future on the enzymes and protein complexes that move these nucleosomes around. And you have people in the audience, Ari Melnick and Sam Waxman talked a bit about it last night, of blocking, say, repressive complexes and transcription factors that would come into these areas. All of this is going to come into play as we consider epigenetic therapy. And I'm going to make a hypothesis to you about how this works. I think of targeting very differently than maybe others do when you come to these therapies. I don't think, and we heard last night from Dr. Waxman, I would say, started out with a specific approach, but it ended up affecting a lot of things. It's a good thing, I think, because I think that we know one thing about targeted therapies. Let's take EGFR mutations in cancer. If you block them in lung cancer, you get a great initial response by blocking the addiction to that signaling pathway. But all too soon, in many cases, new addiction comes back, either because some other part of the pathway takes over or another pathway takes over. So there's a lot of complementation. There's a lot of adjustments the cell makes. All the genes, just the DNA hypermethylated genes I've been talking about, they're all scattered through these Hanahan-Weinberg pathways that are altered to drive cancers. So they're all through here. So what if we really could reverse their effects on these pathways as a program target? You're altering the program of the cancer cell, and this is a hypothesis, such that you would block some of these complementary changes and maybe get therapy or move durability of existing therapies around. I'll talk about on Tuesday that we think we've had a collision that makes us think that priming using epigenetic therapy against two of the things that they show right here, cells avoiding immune destruction, and you're going to hear about this. I know there's a poster about immune destruction at this meeting, or pushing tumor-promoting inflammation by complex pathways, altering these so that the cells become more sensitive to immunotherapy. And Tuesday, I'll talk about that in lung cancer. But what we've done, for the remaining of the talk I'm going to talk about, is to do something. Dr. Waxman said last night these were too broad targets, these agents, at high doses. I agree with him entirely. But I'm going to show you some of the existing agents like Azaytidine, which blocks DNA demethylation, which had been used at too high doses, that if you use it at remarkably low doses, is actually maybe a wonderful drug that may work partially through some of the changes that I've talked about. But even where it's working, we have to prove exactly what changes it's doing. So why do we think about this? Well, it's approved for the FDA, 5-azacitidine, because it is very effective against myelodysplasia. I don't need to tell many of you in this room about that. But the telling parameters of that is how low nanomolar doses probably don't work as cytotoxics. It takes a while for them to work in the patients. It Takes a while for them to respond. And all the pharmacokinetics suggest at a very low concentration to the tumor cells. Can we model this in the laboratory? And I'm going to show you, yes, I think we can. Can we move to clinical trials by taking advantage of these models in the lab? I do think we can, and I'll suggest this to you. And I want to point out something else. The time to patient responses being long, are stem-like cells in the tumor sensitive to agents like this? I'm going to show you that experimentally they seem to be. And the other thing I'm going to show you and remind you is that these drugs, 5-azaytidine, block the catalytic site, the enzyme activity of DNA methyltransferase, but they do something else that people forget about, very important. These low nanomolar doses actually remove DNA methyltransferase transiently from the nucleus because it triggers incorporation into DNA and ubiquination of these proteins. And they have scaffolding functions experimentally. So we have probably something that's dual about these inhibitors that I think actually improves their efficacy. And they will work with histone deacetylases in the lab to help re-express DNA hypermethylated genes. Maybe this is a mechanism in the clinic. Peter Jones taught us about low doses in an experiment in retrospect with Shirley Taylor in 1980, where he took immortalized fibroblasts, treated them with azacitidine, and by this count right here, they turned into muscle cells, micronuclei, as they were lowering DNA methylation. But notice their peak differentiation was way back here in a dose curve where you actually blunt differentiation, even though you're continuing to increase DNA demethylation. And this is probably a cytotoxic range. You don't want to kill a cell you're trying to differentiate immediately. You don't want to kill a cell you're trying to reprogram immediately. So in our stand-up cancer, I'll show you a little bit how we've taken this to the laboratory in work from my lab, but in close collaboration, co-collaboration with a mammary cell biologist and breast cancer biologist, Sidney Zanow, her postdoc, Weili Li, and my graduate student from Taiwan, Sing Tsai. And what we did was try to set up assays for leukemia and solid tumors to work with low nanomolar doses and see how they might be working to give a cell, quote, a memory. So what we did in this work, which we published last April, was to take low doses of Vidaza or deoxyazacitidine or Dacogen and just give it to the cells for 3 days. For leukemia, we usually work at about 50 nanomolar azacitidine, and solid tumors never above 500 nanomolar. Then we rest the cells, and we put them with no further treatment into self-renewing assays of various sorts, which are shown here, feeder layers, nude mice. And we do this in grafting site to the breast or orthotopic. This period here, and I'm going to show you sensitivity for stem cells here. This period here is very important when we rest them because we look at what the treatment did to their genome-wide DNA methylation, expression arrays, and we match these things to find out what's going on. And so you can see that 10 nanomolar of deoxyazacitidine to a leukemia cell can shift the peak of hypermethylation from here by about 30% down to here. And with that, you get re-expression of some of the genes that bear these marks. We have to prove which ones are the most important. But the one thing we learned is that many of these genes, let's take the one in the green here, the drug was stopped right here. The gene keeps going to peak transcription 10, 12, 15 days, sometimes weeks after you withdrew the drug. And I think this is a very important thing that we need to consider in changing gene expression around in therapies. And so when you do this, if you pre-expose those cells, then put them into a mouse, here's a leukemia cell line that only CD34 positive cells form the leukemia. If you put those cells on a renewalator and clone them later, you can completely block that cloning or blunt it severely with 10 nanomolar in these two leukemia lines. You can blunt both the CD34 tumorigenic and non-tumorigenic cells for their methylcellulose cloning. And then if you put these cells into the mouse when they're pre-treated versus not treated, don't treat the mouse, you can severely blunt the leukemia from emerging in the mouse. So a short exposure can really affect tumorigenic stem-like cells. And here are primary leukemia cells done with the same protocol, transient exposure, then into methylcellulose. So you don't affect every tumor, and you wouldn't expect that. Here's an AML that you get nothing from that. But here are AMLs with variable degrees, sometimes profound, again, depression in their ability to clone. And here are primary breast cancer cells from patients that are all luminal or HER2 positive. And you can see they grow in spheres as stem-like cells, with mock treatment, they grow very poorly with transient exposure to 500 nanomolar AZA. You can quantitate that, the black bars here. You can disrupt them, and the treated cells, put them back in the sphere assay again, 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 and they retain this blunting. So there's a memory-type response. And in these studies that we did and reported, we saw all of these pathways harbored DNA hypermethylated genes that were altered by this treatment. We have to prove which ones are most important or what other mechanisms are accounting for this. Sometimes we see signs of differentiation. Usually it's partial. So in this leukemia-treated cell line with 10 nanomolar, when we look 14 days later, we see the more mature granulocyte marker CD11 increasing. We don't see cells that look like mature granulocytes, but we do see evidence of leaving the stem cell compartment, loss of stem-like properties, and gaining of some differentiation properties. Now, we've been turning to solid tumors, and again, I'm going to talk about the therapy trial on Tuesday, but we've taken 8 non-small cell lung cancer lines here. And we know that only a few patients in the trial respond to the therapy alone. And this looks to be the case that you would predict from this. So this is the readout in the mouse. We stop the treatment after 3 days, put the tumors in the mouse, look over weeks at what happened. And most of the cell lines with the AZA alone don't do anything. A few have intermediate responses. But then we see, and we see this in breast cancer, mostly in the luminal type. We see it in this lung cancer. We see it in ovarian cancer. Some cell lines that have a prolonged tumor latency from that short exposure to the drug, taking weeks for the tumors to come up. And we're wondering if this predicts that subset of individuals that respond to the therapy alone, particularly in lung cancer. And I'm going to just leave you with this slide to end, which I'll talk about more on Tuesday. This is a patient who got 5-azacitidine at low doses, has advanced multi-treated non-small cell lung cancer, progressed eventually, and then went on a new form of immunotherapy, which I'll talk about on Tuesday. He's been on that now, you can see, for, I believe, almost 15, now 20 months. He had this big advanced non-small cell lung cancer here, and it looks like this. And I'll talk about Tuesday the fact that the early studies indicate that this immunotherapy, which works about 20% of the time with the immunotherapy alone, is working so far in low studies in virtually all the patients now that the N values are low. And I'll show you on Tuesday the fact that we think the reason this may be is that azacitidine activates a complex system of changes that would increase immune recognition of the cells. Construction of the major histocompatibility proteins on the surface, pro-inflammatory changes which either can push the tumor or have death signals that we see in some of the most sensitive lines, like those 2 sensitive lines I showed you, autophagy signals, viral defense, very complex. And I'll talk Tuesday about a signature that we've derived that we think may very well predict who responds. And I thank you for inviting me. I don't think I'm too much over time, or on time, but I thank you.
Thank you very much, Professor Baylin. This is a very important, comprehensive lecture. Now this lecture is open to discussion.
Hi.
Steve, wonderful work.
Thank you.
I have this question, and perhaps it's not so much a question as a comment. When one's trying to figure out transcriptional targets, one always has to look at early effects, because if you give a patient a drug and it has an effect 6 weeks later, you're comparing cancer versus normal. So of course you see changes in gene expression. So how are you guys going to address this when you're using low doses? It takes several days, so you're really looking at 2 different populations of cells. But you can't really get at the question as far as I can tell.
So Scott, in this therapy, in MDS, it's been well shown by ESA and others, and we see it too. You're not really looking at 2 populations at 4 weeks, because seldom do the patients with MDS change their MDS at 4 to 8 weeks. And so that's why in most of these studies what people do, and they need to be biopsy driven. In the lung cancer trials I'll show Tuesday, we didn't know at the time, we didn't biopsy the patients, but we biopsy at the start and we biopsy at the end of 4 weeks. But the tumor cells are seldom, at least visibly by histology, changed, and the blast cells in MDS in a few patients are actually worse at that time. So you really are looking at the cancer cells, and I think that's something about this therapy that's very different from others. And so if you do chromatin, you do DNA methylation, you can learn a lot in this kind of setting from those early biopsies. That would be my response.
Well, just to give you an example, if you take primary hematopoietic cells from one of our knockout mice, they don't grow at all after you transplant them. But if you put them in culture for 3 days, they now transplant. The reason is in that 3-day period, you selected for rapidly growing cells. So that you're really talking about a different population after just 3 days. So I would suggest that after 4 weeks of treatment, you are looking at a different population of cells.
No, I agree they could be a different population of cancer cells, but maybe that's what you want. I don't think what you're looking at is normal cells, because if you look at the markers, you look at the blasts, you look at all of this, that's not what you see.
They will
So you want different cells, different cancer cells. That may be all right.
Yeah,
but I agree with you.
I'm trying to get to the point of how do you find the actual targets?
Well, I'll show you on Tuesday what we found in the immune system is some upstream transcription factors that are hypermethylated, where you can get signatures even in the primary tumors where low expression, loss of that transcription factor accompanies a lot of loss of downstream events. And this is one thing we're excited about. This may be the way to tap into it, because you're getting pathway changes. Yeah. But I think this is the way to go after.
Forward to that.
So Steve,
yes.
Steve.
Hi, Ben.
How you doing, Steve?
I'm sorry.
Is this on?
Yes, I think
Okay.
so.
Do you know the status of the DNT mutations, TET and IDH mutations in the leukemias that do or don't? Oh yeah, it's not working. Do you know the? Oh, there it goes. Do you know the status of the DNMT, TET, and IDH1 mutations in the leukemias that do and don't respond?
From what we know, who others have sequenced, but we need to sequence our leukemia lines. In the lung cancers and the work where we're getting efficacy, I'm not aware of any DNA methyltransferases. They didn't see those in the cancer gene. In the leukemia cells, to my knowledge, but Ari may be a bit, do you know the sequence for Kasumi, say, in KG1A?
No, I mean the primary leukemias. You showed
Oh, no. And that's another, I agree. The genetic background here and the different therapy responses to this, Ari and others, is a very important area, and I agree. Yes?
Professor Xiong.
Hi, Steve. So the question is regarding the repression of P16 by either polycomb, which
Yes.
is on histone methylation versus DNA methylation.
Yes.
So what you described in this Child Pro study in the AAA negative breast cancer versus the luminate type. So looks like the P16 is repressed either by DNA methylation or by polycomb that appear to be mutually exclusive.
What I showed in breast cancer was not specifically for P16. I showed you hundreds of genes, occasionally P16, but those were not specifically P16. There were many other genes that were more methylated in the luminal than and Tim Chan, several labs, a group in Sweden, they've all found this to be the case, but not P16 so much among that.
All right. So then my question can be modified. So when you present the TCGA database,
yes,
where the P16 mutation and the DNA methylation look like occur in a mutual exclusive manner, but the P16, when cells are in the early stage of development or at a young adult,
absolutely,
they are repressed primarily by the histone methylation through the polycomb. So my question is, are DNA methylation and the polycomb-mediated the chromatin repression, are they linked or they are really alternative way of silencing P16?
This is a great question. I'm glad you mentioned it because I always forget. So P16 is a classic example. If you look in normal tissues, very low expression. So you would think, well, what difference does it make to silence it? But if you challenge those cells, send them into senescent, it has to be induced. P16 comes up. For many of these polycomb genes or bivalent genes that look low in normal tissues, the question is this. They're still available. If that cancer cell can't turn them on, that may be a bad thing. DNA methylation is much harder to turn on than polycomb or bivalency. So the question is, are a lot of inducible genes that need to change to kick a cell out of self-renewal affected by this? And I frame it as a question, but it's one of the leading questions in the field, actually, to really look at that very question.
So you are implying that DNA methylation has been evolved to replace the histone methylation during the...
Quantitatively, yes. It's not a complete loss of polycomb mark, but Peter Jones and Jean-Pierre Issa call it switching. I guess that's okay. Yes, you're switching in some way. Correct.
This might be a naive question. So from your TCGA data, you show a lot of global changes, epigenetic changes. Let's see if you can derive an index from your global epigenetic changes, for example, certain methylation level. Do you think this can somehow predict the prognosis of your cancer? In a certain way, in your epigenetic regulation, I think it's an essential topic, the specificity, right? Do you really think the global level or extent of your changes is more important, or some modifications on certain loci of methylation or whatever acetylation changes is more important?
So you've asked a couple of complicated questions here. Let me give a hypothesis to you. I think from the work of Peter Laird and Berman and others, and our own work and other people's work, what I think happens for a lot of these genes is you're shifting chromatin boundaries around in the cancer cell. Mutations may be involved in some of them. You're losing methylation, you're gaining. So not all the genes are being specifically targeted, but you're catching up a whole bunch of genes in this change. And the question is, we know some of them are tumor suppressor genes. They're important. Others in groups might affect developmental biology context, the way I was talking about. So no, I don't think everything's specifically targeted. I think it's a program of changes. And within that program, we need to prove what are the important genes, or I think groups of genes. Because if 10 developmental biology genes are changed so you can't kick the cell out of self-renewal compartment, that could be enough to cause a contribution to tumorigenesis. But that's a hypothesis, that's the question.
Yes. When you look at blood cell, normal cell differentiations at a very primitive stage, they give all kind of capacity to differentiate.
Correct.
My question is versus at the epigenetic program level. At the epigenetic program level, you think you see from normal cells shift to potential cancer cells, so the change will be potential of the first change will be greater or smaller? I mean, gradually gain the potential or
Right.
gradually reduce
So
the
what I'm
potential?
hypothesizing to you, and remember, I showed you just one epigenetic change, that you're diminishing the potential for those cells to leave an earlier compartment. Not my work, but we know from all the beautiful data on doing the epigenome in normal embryonic stem cells, they need to switch these marks around to leave the stem cell compartment, go to the progenitor compartment, go to the differentiation compartment. I'm saying the cancer cell may be repressing and locking down some of that machinery, among other things it's doing. But this change I was postulating might affect that, damp that totipotentiality down and leave them more self-renewing and differentiation blocked.
Thank you.
I see. Yes.
I want to thank you for that, Steve. It was very illuminating.
Thank you.
The role of the bivalent gene population seems to me to be a great target to understand that stoichiometry,
Yes.
whether it gets active or silenced. And I would think, and you probably showed it, but I'd like you to emphasize it, clarify it, that would be much more of a target in cancer cells than in normal cells, because characteristically a cancer cell is very adaptive.
Yes.
I mean, particularly to therapy, and as you talk about that. And we know some of those genes that are involved in drug resistance and transport. So I would say that if I was going to look at this, I'd go by function and therapeutic goal. So, and I know you're going to talk about that. So I would think that the cancer cell in general has bivalent set of genes that are related to drug resistance that will be readily turned on and activated, and that could be prevented by azaytidine. So the stoichiometry of changes, I think, are critical in studying this. I'm wondering if you've addressed that yet.
I couldn't agree more.
Yeah.
Of all the bivalent genes in cancer, or in embryonic stem cells, remember, most of them don't take on this DNA methylation change. It's a subset. It's hundreds out of a couple of thousand. Stu Orkin and Weinberg have said that there are a number of polycomb genes that don't come up normally. We've looked at this. So a subset of those have gained DNA methylation, but not all of them. So there are many mechanisms controlling this, and I agree with you. You have to know that biology, and probably within every tumor type, you've got to map some of these things. Yeah, so it's not all the bivalent. And the last thing I'll say is if you do conformational studies, we've just done one team intensely, You have one kind of looping going on around bivalency. If that same gene is DNA methylated in a cancer, it changes those loops. It loses the polymerase. It's much more silenced than it is when it's bivalent.
Thank you very much, Professor Baylin, for very interesting work.